Okay.
Good morning.
Hello, everyone. Thanks for coming to this event. This is the first workshop of a series of 4 workshops organized by the partnership between the TWRA, that stands for Tropical Water Research Alliance, and RMIT, the Royal Melbourne Institute of Technology. Today, we will hear from 4 experts about the research on microplastics. In our panel of experts for this event, we will have 2 researchers from RMIT and then 2 researchers from the TWRA that I will briefly present to you very soon. We will have the event as this. So we will start the event with the 4 presentations, and we'll have questions at the end. So please feel free to send your questions to us at any time during the event. So we will start with a presentation from Dr. Biplab Pramanik. He is a senior lecturer in civil engineering and infrastructure engineering discipline and a member of the Water Effective Technologies and Tools WET Research Center at RMIT University in Australia. He is an ARC TWRA Fellow, and his research focuses on the development of separation-based technological platforms for removing emerging pollutants, such as nano and microplastics from water and wastewater. And his current research also focuses on fatberg management and resource recovery. This presentation will be followed by Dr. Jack Myers. She is a senior research fellow with RMIT University's AquaQuest Research Group. She has worked as an aquatic ecotoxicologist over 17 years experience in this field. Her research interests are focused around marine and freshwater ecotoxicology, particularly on understanding how human-induced stressors affect fauna and flora in aquatic environments. And recently, a major focus of her research work has been on understanding litter in the environment and supervising students in this area. Then we're gonna hear from Dr. Cristina Branco. She's a full professor at the Federal University of the State of Rio de Janeiro in so-called UniRio for us here. She's coordinating institutional projects in the area of biodiversity, including limnology of coastal lagoons, hydroelectric reservoirs, taxonomic and ecology of freshwater plankton, diet of neotropical fish, and biological indicators of anthropogenic impacts. She is a leader of the Biodiversity Research Group on continental aquatic ecosystems and surrounding areas. And our final presentation will be given by Dr. Uwe Schulz. Sorry for my pronunciation. He is a full professor of biology and engineering at the University of the Vale do Rio dos Sinos, Unicinos. His research interests are focused in ecology, with emphasis on aquatic ecology, working mainly on environmental impacts on water bodies, environmental education, fish migrations, and fish radio telemetry and fish habitat use. So this is our panel, and I would like to invite Dr. Biplab Pramanik to start his presentation.
Thank you. Thank you for the nice introduction. Can you see my screen?
Yes.
It needs to go to presenter mode.
Yes. Yeah. How about now? Is that clear now?
Yeah, right. Perfect.
Okay. And thank you, and good morning to who joined from Australia, and good afternoon to Brazil. And my talk is about fate control of nanoplastics and microplastics. I've been working on this from last just 3-4 years. So I don't have a huge experience, but from last couple of years, we have number of PhD student and research fellow, they work on this. So I do have some update on the topic. If we talk about the microplastics, I maybe don't need to define because you all know. And this is kind of a plastic fragment, which are usually less than 5 millimeter in size. And they are hydrophobic in nature. And another one is they are resistant to thermal and biodegradation, and their density vary from 0.8 to 1.6. Sometimes it can be more as well. When these densities mostly for the pristine plastics or microplastics. If we have other element deposit on the plastics that we found the density go up to more than that, it is the reason there are a lot of microplastics they deposit on the sediment. So, and there are a number of type of microplastics we have, and commonly used they are polyethylene, polypropylene, polystyrene, PET, PVC, and synthetic rubber. Somehow, when we see the microplastics from different type of sample, these are the major sources we found. And another one is when we have done some work related to risk and other things. I have not done much work related to risk, but we found from the literature and which are linked with our work that they work as a harmful carrier for the toxic chemicals. We understand that in typical microplastic sample collected from wastewater or road dust or whatever, we do have a huge amount of heavy metals, and sometimes, in fact, the PFAS they deposit on the plastics, microplastics surface. So when it goes to the water environment, it is a problem. So then we can consider it is one of the highest carrier for harmful of toxic chemicals. In fact, when we talk about the nanoplastics, they are more, harmful compared to microplastics because of the surface area. Then easily they can absorb or PFAS and other heavy metals and organics, they can deposit on the nanoplastics. And eventually it goes to the environment and has a lot of consequences. So we have done some work related to, this was in fact 3-4 years back, this was my first work. So we tried to understand the source of microplastics from road dust. And we understand that if we look at this figure, we see that there are a number of things. For example, tire, these are the thermoelastomeric polymer we use for the marking, and plastics around the roadside. And when we have this is from the road dust, and when we have heavy rainfall event, eventually it goes to the water environment through the storm water. And there are a number of researchers, particularly this number, like 42% from the tire, road marking paint, around 70% household, from 90%. These are the number we got it from Norway Water Research Institute. In fact, they have started working on microplastics a long ago. And what we understand here that tire is one of the major component. TWRA is one of the major component of microplastics in road dust, and eventually it goes to the stormwater, and then in the water environment. We try to collect sample from different part of Melbourne, western and southeast part, from different area, for example, residential, industrial, commercial, and we understand that in the industrial area we do have more microplastics particle in the road dust sample compared to the residential as expected. And the major microplastics that we found in the road dust sample, they are polyester, nylon, uh, LDPE, PP, and polyester was the maximum one, and the fiber dominant in the road dust sample, which was around 47%. Other are fragment, braid, and film. This is kind of a overall generic information because we have collected sample from more than 50 different sites. And at the same time, we try to understand uh, what is happening with the stormwater sample. And again, we have collected from different places, from wetland, including the wetland. And we understand that uh, in industrial area, we have around 26 microplastics particle. Residential area is little bit lower than the industrial area, and almost the similar type of microplastics we have in the stormwater. It means like road dust sample, road dust microplastics is going there, and PT was one of the major polymer, and dominant shape was fiber, same as road dust sample. Another things we realize here when we have done the particle size distribution, stormwater sample has a lower, similar type of plastics we found, microplastics we found, but the size was a bit smaller because of what we feel that this is mainly because of maybe water shear force, they break down the bigger to a smaller one, and this smaller is always a little bit concern, I always feel. And at the same time, we collected sample from wastewater treatment plant from local water utilities. I can't tell you the exact water utility name here. Still, we are working on with them. So we understand that in here, the way we design the drainage system is bit maybe different than the Brazil. We don't know about your system. Here we have a different drainage system for stormwater and municipal wastewater. And municipal wastewater source, I think here, if you see the figure, you see that industrial domestic process. And if we talk about the microplastics, mostly they're coming from textile fiber, from washing machine and plastic commodities, particularly personal care product and cosmetics, mainly from the toilet and other places. It goes to the wastewater treatment plant. And we collected sample from 3 different treatment plants in Melbourne. And they are over the time we collected just to understand the seasonal variation as well. And here we found that the synthetic fiber was one of the major one, followed by the fragment. And most of this, if we talk about the size, the majority, more than 50%, their size was between 500 to 800 micron. And the major polymeric materials was polyethylene, PT, polystyrene, and polyester, to some extent the same as stormwater. And average, we found around 42 microplastics particle per liter. And another one, what we understand that in the summer season, we do have a more microplastics in the influent of the treatment plant compared to the other season, because I think summer we use LOND more than the other time, maybe. It is the reason we have more fabric and other things as a microplastic in the treatment plant. And a good point, what we realize, and we highly appreciate water utility, because all the wastewater, because 3 wastewater treatment plant, we see that they can remove up to 98% of microplastics. But still, if we consider the total volume of water, and this 2% contribute a huge amount of plastic to the downstream environment. But overall, 98% removal is a good one. So another things we realized that because we collected sample at different phase of the treatment plan, and primary treatment plan alone can remove more than 60% of the total microplastic. Now, what we understand that, and our concern is, we said that 98% they remove by the wastewater treatment plan, and how those remove. What I feel that they remove because they mainly goes to the biosolid, and particularly in the primary and secondary treatment process. So if we can, and then it goes to the tertiary, and we do have most of the cases membrane and other things to remove. If we have the RO, they are very good to remove. So ultimately 98% remove, but it goes to the biosolid. And if we use this biosolid, and definitely EP and they have the restriction of using biosolid, and there are a lot of water utilities in Melbourne. I know they are working on how we can, like treat the biosolid and can use for different application, including Southeast water, because I have been a part of number of projects with my colleague Kalpit with Southeast water, and they have been doing a lot of work on biosolid management. In that case, even we found that we are using pyrolysis and somehow, and the pyrolysis can remove the microplastics from the biosolid. Is this good? If it is not, and then if that biosolid is used for different application, we will be in trouble. So based on this experience, what we understand that if we can do something at the very early stage in terms of the removal, that would be good. Okay, this is about identification. And look at here, if we talk about a typical wastewater treatment plant, most of the cases we have primary, secondary, and tertiary treatment unit. Because the concept here, we got it from stormwater sample, because what we understand that from road dust to my stormwater, when it goes because of water shear force in the drainage system, the size is getting smaller. So then we understand that in the wastewater treatment plant, we do have a lot of mixing, pumping, bubbling, this kind of facility, and they generate up to 150 kilojoule per liter of water shear force. Then our hypothesis was, okay, if we have this kind of water shear force within the wastewater treatment plant, we understand that they remove microplastics. Are they generating nanoplastics within the wastewater treatment plant because of high water shear force? Then we have done some experiment just to understand the fragmentation of microplastics to generation of nanoplastics. And we have simulated the mixing process at different, like as we see that we have a water shear force up to 150 kilojoule per liter from the reference, and we use up to 100 kilojoule per liter. We understand that a huge amount of nanoplastics generate within the wastewater treatment plant. For example, when we are using 32 kilojoule per liter of water shear force, and it increases around 28 times of nanoplastic. So again here, if we can do something at the very early stage to remove microplastics and to make sure microplastics not breaking down to the nanoplastics, that would be a very good outcome. Another thing here, I realize and I believe that, and we talk about wastewater treatment plant can remove 98% of microplastics or 99% or whatever, because somehow there are a lot of, and the system, microplastics goes to the biosolid at the same time. There are a lot of microplastics that transfer to nanoplastics, and identifying nanoplastics is a bit difficult, and we are not considering this one in our count when we are considering that one. It is the reason that 98% or 99%, that number may be not that accurate, because they break down to nanoplastics and we are not considering that one. In our work here, so we use nanoparticle tracking analysis. As we said, that identifying nanoplastics is a bit difficult. We are trying to develop method on that at RMIT University. And here, this work we use like a simulated water environment, and that can, an NTA, nanoparticle tracking analysis, is good to use for, clean type of water where we have only the nanoparticle and nanoplastics and microplastics. And the problem is when we have the real sample, it would be very difficult, but we are trying to do that one in our work. Okay, then we try to use treatment process, and flotation, dissolved air flotation is commonly used at the primary treatment of the wastewater treatment plant. Particularly, the purpose is a bit different, not for the microplastics. So they mainly used for fat, oil, and grease removal, and suspended soil, other things. And just we are trying to understand if they can remove microplastics or not, and they are really good, because when we are using the surfactant in the flotation process, they can improve up to different type of microplastics at different concentration they remove. So overall, we are having a good removal efficiency when we are having the air flotation process. So I feel that if we can do something at the flotation unit in the treatment plant, because this flotation unit, the way we use for treatment plant is not for microplastics, but if we can redesign on, particularly if we have the bubble size a little bit smaller, so that we can have more, like attraction of nano microplastics, that would be good way to go. This way will help us this will help us to go microplastics go downstream to the next stage, for example, sludge. And we also use the coagulation process, but the removal efficiency was not that really good. But when we are having, like whether microplastics or our microplastics, removal efficiency was much better, but not for the pristine one. And as we said, that membrane is always used at the last lot of cases if we want to use for different application. And they're very good to remove. They remove 99.6%, 91% microfiltration and ultrafiltration. But the problem is when we have a different size of microplastics, their filtration performance is a bit different. Removal efficiency it was okay, more than 90%. But the problem here we found is the fouling with the commercial membrane ultrafiltration and microfiltration. And I got ARC project last year, fellowship project. It calls Australian Research Council Discovery Early Career Fellow. So it means like next 3 years I'll do only research, and the project is about to develop a novel membrane to remove nanoplastics. And just I have started from last 2-3 months. We don't have much results. So we synthesize membrane at RMIT University, and we have done some test it, and we understand that the commercial membrane is a major problem. They can remove, like if we optimize, they can remove more than 98%, 99% or whatever. The problem with the water flux that we need to make sure and keep in our mind. And here the MOP-based membrane, metal organic fed membrane, they're very good for removing. Complete rejection, we can get it here. At the same time, we can have around 7 to 8 times more water flux than the commercial membrane, because the specific surface area is extremely high for here. And we have done some tests like FTIR, XRD, and other tests to understand that whether our synthesis MIL 101 chromium membrane is accurate or not, and the peak is well aligned with the standard. So our synthesis was right. So yeah, this is all about my work so far on this one, and happy to respond to any of your questions. This is all about for me. And from last couple of years, we published around 15 paper journal articles on the topic, and many of them are from my PhD student, and I have done some work alone as well. So if any of you need any information, feel free to ask, then we can send this document to you guys. And we acknowledge SPEL was my first industry partner to work on microplastic in stormwater. And then we have some funding from CSRO and ARC. I did not include one of the water utilities here because they said don't disclose all that details. So if we acknowledge them, then you understand that we take sample from them at this stage. Yeah. Okay, this is all about for me. Thank you.
Thank you. Thank you, Dr. Biplov. I would like to invite Jackie to give her talk now, and then we will do the questions at the end. Thank you.
Hopefully that's sharing with everybody.
Yes.
Great. Thanks, Vanessa. Welcome, everybody. So yes, I'm Jackie, and I work with the Aquatic Environmental Stress Research Group here at RMIT. And today I'm going to just give you an overview of the research that our group has been undertaking, looking at litter and microplastics in Melbourne's waterways. I would like to say that the research that I am presenting is mostly undertaken by students in our research group, so not my own work. Okay, so our research group started working on litter and microplastic related research around 2018. We've just completed a large project looking at trying to develop methods for monitoring macro litter in freshwater environments so that we can start to understand catchment conditions in relation to litter and start to make targets to manage and reach for macro litter, and that's work I've been leading. We've also completed quite a few projects looking at microplastics around Melbourne, and these have been focused on two main areas of microplastics, and that's their types and levels in waterways across the Greater Melbourne area and on their occurrence in biota and the potential for biota to be used as biomonitors of microplastic pollution. So I'm going to focus on the microplastic research for today. So what are the types and levels of microplastics that we see in Melbourne's waterways? So this was a first study that we undertook. It was in 2018, and it was looking at the association of microplastics and land use across Greater Melbourne. And so the study investigated microplastic pollution in sediments of urban wetlands, and we also mapped land use around those wetlands and then tried to look at the associations between the occurrence of microplastics in the wetlands and the land uses. And what we found was that microplastic abundance in sediments, it varied across the wetlands, ranging from 2 up to 147 microplastic items per kilogram of sediment. Now, there was a dominance of fragments at sites in this study, followed by fibres and beads. And interestingly, there were only 7 sites where one plastic type was found, so either fibres, fragments or beads. And what this sort of suggested was that there could be specific sources of microplastics to those particular wetlands. There was a strong association between urbanisation and microplastic pollution. So where there was a higher abundance of microplastics, this was where there was increased urbanisation. We also found a relationship with industrial catchments. So where the catchment area was more than 15% industrial, we had higher levels of microplastics. From this early study, what we sort of thought was the dominance of fragments suggested that the source of microplastics is predominantly secondary, so those formed from macroplastic litter that's entered the environment. In a follow-up study, we looked at a number of different types of waterways across urban and rural areas. So we looked at estuaries, streams and wetlands. And in this study, we collected both water and sediment samples and assessed them for microplastic presence, form and polymer type. Now, in this study, what we found was that microplastics were detected in 95% of water samples and 96% of the sediment samples. There was no difference in the microplastics abundance between the different water body types. The abundance of microplastics was higher, though, near the coastal cities than in the less densely populated inland areas. And again, commercial, industrial, transport-related land uses were associated with an increase in microplastic abundance and also in polymer diversity. Now, in this study, fibres were the most common shape that were found in both water and sediment, and the percentage of fragments in sediment was up to 3 times higher than that was found in waters. Now, there were 14 different polymer types found in water and 19 in sediment in this study, and polyester was the most common type, which is very similar to what Bitlop found in their studies. The next piece of work we did around microplastics actually was looking at microplastics in roadside dust. So this was looking at rural and urban areas across Victoria, and we wanted to see what level of microplastic contamination there was in background dust and also try and determine possible sources. So we looked at 16 locations, and we looked at 2 different seasons in this study. Now, the average microplastic abundance over the 2 seasons ranged from around 21 to 529 items per kilogram, but there was no difference between the 2 seasons. The highest abundance occurred in the Melbourne area or in regional areas where there was a high level of urbanisation. Now, again, fibres were the dominant shape, followed by fragments and film. And at 3 sites, we found pellets that were only found at 3 sites. In this one study, polyester, polypropylene and rayon were the most common polymer types. And what we found is like urbanisation and rainfall had an influence on microplastic accumulation in this study. What we have noticed though is microplastic pollution levels are kind of considered low in the studies that we've got when you compare with similar international studies. But when we look at this study compared to those previous 2 studies, the levels that were detected in the dust were higher than what we found in the wetlands. Currently, we have 2 PhD students that are working with us looking at microplastics. And the first one is by Eamon, and he's looking at the efficiency of constructed stormwater wetlands at capturing macro and microplastic pollutants. So he's trying to look at the relationships between macroplastics that are occurring in wetlands and microplastics, and try and understand constructed wetlands kind of a source or a sink from microplastics. So are we seeing lots of macroplastics which then form microplastics, and these move through the wetland into waterways, or are wetlands actually capturing and retaining these? And he's in his 2nd year of his study, so he'll hopefully have some work coming out soon in publications. The second student has just started with us, and she is looking at the sources, fate and transport of microplastics in a really large coastal embayment close to Melbourne. So as part of her study, she'll be trying to understand if factors like land use influence microplastic occurrence. This is a really agriculturally dominated catchment. So trying to understand if different agricultural activities lead to different levels of microplastics, but also trying to understand whether other factors like maybe sediment type and that influence the transport and fate of microplastics in this system. So now I want to give a brief overview of a couple of studies that we've undertaken that have looked more at either impacts or occurrence of microplastics in biota. So the first study that was undertaken by a PhD student was looking at microplastic uptake in a noxious fish species called the mosquito fish. So in this study, fish were sampled from 9 wetlands across 2 regions within the Greater Melbourne area. And the amount and types of microplastics were investigated in the body and the head to assess different uptake pathways. So the head kind of thought of representing gill uptake, and the body ingestion of microplastics, and also looked at how body size and sex also microplastic uptake might differ between those as well. So what the study found was that the bodies of fish contained more microplastics on average than the heads. And also the size of microplastics detected in the heads was much smaller than those found in the bodies. So microplastics are probably more likely ingested compared to being uptaken from the gills. Microplastic uptake was proportional to size and weight of fish. So larger fish had more amounts of microplastics in them than smaller fish. Also found that female individuals had a greater tendency to ingest more microplastics than males. The dominant type of microplastic was fibres, and the dominant polymer type was polyester, although there were 11 different polymer types that were detected in the fish. And what the study really showed us is that microplastics are taken up by fish, but the abundance is different depending on the uptake route, the size, and the gender. In comparison, though, to similar global studies on microplastics and biota, these results indicated a relatively low prevalence of microplastic pollution in at least this fish species from the Greater Melbourne area. Another study undertaken by, again, a PhD student, investigated microplastics in the Australian glass shrimp. You can see it up in the right-hand corner up there, and also looked in surface waters collected from rural and urban areas across Victoria. This study, they wanted to understand the occurrence of microplastics and the types in water and in shrimp, but also see if the shrimp could be used as like a bioindicator of microplastic pollution. So microplastics are present in both surface waters and shrimp at all sites. So this indicated that microplastics are ubiquitous and occur in both rural and urban environments. 36% of shrimp contain microplastics in their bodies, while 100% of the water samples contain microplastics. The dominant form of microplastic found was, again, fibres. More forms of microplastics were found in the water than in the shrimp. However, it was found the shrimp ingested a greater variety of polymer types. And shrimp seem to have a preference for blue plastics, even though there were other colours available in the water. And this ingestion of just blue fibres is possibly shrimp mistaking them as like a food item. There was no relationship between shrimp size or weight and the number of microplastics that were taken up. But this student also did some lab studies and found that the shrimp retained microplastics in their gut for between 12 and 24 hours, with some individuals still containing them after 48 hours. So what they sort of thought from this was that the shrimp could be providing a picture of recent microplastic pollution at sites. I just want to kind of sum up the work that we've been doing and just sort of think about some of the challenges around microplastics. And I suppose one of the key things we found was that, and similarly in Biplop's studies across Melbourne, is that fibres are the dominant form of microplastics in most of the studies, and polyester seems to be one of the most frequent polymer types that we're finding. Urbanised water bodies, particularly those surrounded by commercial and industrial land uses, tend to be more polluted with microplastics compared to other areas, and Biplop found this similar to that as well. When comparing results with similar international studies, though, the level of microplastic pollution in our urbanised areas is probably a lot lower. We found that biota does have a potential as a biomonitor of microplastic pollution and can be used to provide an assessment of the amount of microplastic loads discharged from rivers to potentially oceans. One of the key sources is likely to be macro litter. And so managing and reducing macro litter could be really significant to reducing microplastic pollution, but there is more research needed in this space. And the other thing is that urban stormwater management, like in that area, we focus a lot on contaminants like dissolved things like nutrients and metals. And there's a lack of thinking about microplastics in stormwater. And so I think there does need to be a better handle on urban stormwater management in terms of microplastics. I'd like to thank you all for listening to me today and joining us. I'd like to thank all of the people who have contributed to the research I presented today. One quick plug too, though, in a different stream is I do have a PhD student currently looking at pesticides using passive samplers. And she is based in Italy and is looking for community groups that might like to come on to be a part of her project to look at pesticides in local water bodies. So if you're interested in learning more about that, please feel free to drop me an email. Thanks.
Awesome. Thanks a lot, Dr. Jackie, for your presentation. And I would like to invite Dr. Christina to give her talk, but also remember our audience to please send your questions here or on YouTube. Send it to us and we're collecting them all. So please, Dr. Christina. No. Dr. Christina.
Okay.
You're here?
I'm coming. I'm coming.
Oh, thank you. Yeah.
It's okay. Full screen.
Full screen. Yeah, maybe you should click that. Yeah, perfect.
Yes.
Yes.
Perfect. Well, hello to everyone. We are going to present the effects of polymer micropellets on cladocerans and the plankton versus microplastics in a tropical polluted river. First of all, I'd like to present our group. We are part of a tropical limnology group. We work at the Federal University of the state of Rio de Janeiro, in Rio de Janeiro, Brazil. And our group is comprised by 4 professors, 1 technician, 1 postdoc, 3 doctorate students, 2 graduates, and 11 undergraduate students. And our team is involved in research on the ecology of freshwater plankton, trophic interaction in aquatic communities, water quality in rivers, and here including the microplastics, lakes, reservoirs, and coastal lagoons, and environment and human health. Well, the microplastic team, it's a group inside a large group. It's comprised by me and Professor Samira, the chemical engineer Marcelo, and the 3 doctorate students that are involved in the topic with microplastics. So it's a challenger. Our concern is plastic everywhere. In our daily life, we are producing plastics, releasing plastics in our activities daily. And our main concern are the microplastics, size between 1 micrometers and 5 millimeters. And our great challenge is trying to find the relationship between plankton and microplastics. The phytoplankton and zooplankton have a very nice and very good connection in natural system, but things changed by the ubiquitous presence of microplastics. So we have some targets. And the first one, cladocerans and microplastics through experimental research. The second, presence of microplastic in aquatic ecosystem across rivers, coastal lagoons, and reservoirs, mostly field research. Microplastics and water for consumption, and microplastics and human health. Well, the first one, we have a special organism that is helping us to see a lot of things between species and microplastics. These are small cladocerans, Moina micrura. It's a well-known cladoceran. It's distributed in tropical and temperate region, and it's being increasingly employed in laboratory trials, toxicological studies, and is easy to cultivate at laboratory conditions. So we have some results that were published 2 years ago regarding the uptake of microplastics by a tropical freshwater cladocera, revealed by the polyethylene terephthalate fluorescence. Our target in this study was to know if the Moina could ingest the microplastics. So we produced PET prepared by grading pellets. The PET structure was confirmed by the Fourier transform infrared spectroscopy. And the PET particles had different sizes and heterogeneous shapes as the particles that are in the aquatic environment. the same size of the phytoplankton food for the cladocerans. And the blue emission of PET, the natural blue emission of the PET, was observed under a microscope with UV. And in our results, we can see here, in the first 2 hours of experiment, the cladoceran only receiving microplastic, they accumulated in the digestive tract all the microplastics in the environment. The microplastics were seen in the thoracic appendages, and in the molten exudes of the cladocerans, we can see full of microplastics. And the neonates and the juveniles that were born during the experiments show the gut containing PET microplastics within 24 hours. And amazing and very sad, we have here, we can see the efforts of the cladocerans trying to empty the gut full of microplastic without success. So the microplastics were retained in the gut of the cladocerans. Our second target, the presence of microplastics in aquatic ecosystem. We have now a field research in a very polluted river. This research is part of a doctorate thesis of Everton Fitterman de Oliveira. Plankton, microplastic, and water quality in the Guandu River. It's a very polluted river in Rio de Janeiro. This project is inside a large project, the project EcoShift the Guandu River, coordinated by Professor Luciano Santos, and with financial support by several institutions, and supervised by B and Professor Samira. Guandu River receives waters. Here is the basin of Guandu River. This river receives waters diverted from a large river, Paraíba do Sul River. And the river basin, it's a very complicated one because there are several municipalities that don't have domestic sewage treatment. And so the Guandu River receives a large load, a high load of nutrients and untreated sewage, including plastics. And we have in this Guandu River a large treatment plant, the Guandu Water Treatment Plant, which provides water for several municipalities, including the city of Rio de Janeiro, and supplying water for approximately 9 million people. The methods of this research, we have already conducted 2 sampling at rainy and dry season at 12 points around the stretch of the river. We measure physical and chemical variables, including total carbon and nitrogen analysis, and studying the phytoplankton, zooplankton communities, and studying richness, abundance, and biomass of both communities. And for the microplastics, we use nets and sieves with 3 meshes, trying to reveal size diversity of polymers. So we studied the size, shape, density, and type of polymer by UV microscopy, as well trying to find within zooplankton species the presence of microplastics, and analyzing by FTIR the type of polymers present. And we have some preliminary results, first of all, the community, the plankton community, is typically of a very eutrophicated system. We have species of zooplankton and phytoplankton, very common in eutrophicated rivers. And we have the presence of microplastics, especially fibers, in the water of the system. So, our third target of our team, microplastics and water for consumption. We have now ongoing experiments regarding water for consumption for the people of Rio de Janeiro. And we have also ongoing tracking scientific knowledge increase. We have to do this every week on the effects of microplastic on human health. So many things to do, a lot of work ahead. Thank you for your attention.
Thank you. Thank you, Dr. Cristina, for your talk. I'd like to invite Dr. Uwe to give his talk.
Okay. One moment, please. Are you seeing my screen?
Yes.
Perfect.
Yes.
Yeah.
Okay, thank you. So, good morning, Australia. Good evening, Brazil. My understanding of our event was, since we only have 10 minutes to present, that I'll give you a general overview of our situation, of our research projects, of our environment, and of our research group. So I will be talking about microplastics in the Sinus River Basin in southern Brazil. This is the main subject. But I shall be talking about where are we? So giving you a geographical orientation about our research area, what is our environmental situation, and then I'll be talking about some research projects concerning microplastics in raw and drinking water, and microplastics in fish, intestines, muscle, and liver tissues. So where are we? Well, Brazil, it says on map A, and our state, Rio Grande do Sul, is the southernmost state in Brazil, which is shown on map B. And map C shows the location of our water basin. So this is on the left side, on the right side. The whole area is about 4,000 square kilometers, and the main river, which is the Sinus River, has about 190 kilometers length. And the flow direction is from east to the west. And the dots which you see on the map indicate the water quality of the Brazilian scoring system. And you see this is full of red dots, and red dots means water quality class 4, which is the worst one on our scale. By law, this water should be used for drinking water, but most water of our water basin comes from these areas here. So the headwaters are in very good condition. This is pristine rainforest still. But, and here we take our students for snorkeling, and this is an environmental education course. It's a kind of fun class we have. Everybody likes it, and here we have the environmental conditions to do that. But if you go downriver to the lowland parts, you'll see that the urban areas are approaching the river, and you see some large wetland areas still present. This is in the municipality of São Leopoldo, where my university is located. The most severe problem that we have in our water, in our river basin, is untreated sewage. It is, like my colleague from Rio de Janeiro already told you, we calculate that about 15% of the total sewage amount is treated, which means that 85% are not treated. And this is the way it is discharged into the main river. Well, you see completely untreated black sewage entering the Rio dos Sinos main river stem. And about 1 kilometer downstream of this discharging point, there is a station which pumps the drinking water out of the river. So this is a very severe situation, basic sanitation problems. This graphic shows a river gradient coming from the headwaters on the left side to the river mouth, showing the coliform concentrations and counts, and every bar is an annual mean. So if you look at this figure here, there is a red dotted line in the middle, which is the limit of class 3. So all these bars exceeding this dotted red line put the sampling points into class 4, which is not appropriate to the level of drinking water. Right. So this is the main problem of our watershed. And then within this problem, within this panoramic problem, we decided that it would be a good idea to look at microplastic concentrations. And the first project we conducted compared the concentrations of microplastic particles in raw water of the Sinos River and in drinking water, which was derived from this raw water. And on the right side, you see our watershed again. The blue points are the sampling sites of the raw water, and the red points are the sampling sites of the drinking water, which were taken in the residence. Table 1 shows you the distribution of the particle shapes. And as most of my colleagues already found out, fibers are dominant. They are dominant in about 90% of the raw water and 80% in drinking water. And so the distribution between raw and drinking waters concerning fibers, pellets, and films was not significantly different. On the left-hand side, the results of the comparison of the particles in drinking water and raw water. We have almost 1/3 of the particles which were encountered in raw water in drinking water. So this means that our raw water treatment is not very efficient. It seems that about 30% of the particles encountered in the raw water, they pass. The figure on the right hand shows particle concentration on the sampling sites. On the left hand are the sampling sites with a low urbanization gradient. These are the headwaters on the left side. And on the right side are the urbanized areas. So to our surprise, we found a lot of microplastics in the headwaters. And then it seems a kind of removal process occurred. This might be sedimentation, and then a new input in the urban areas. So this was a surprise because we thought that at least the headwaters would be relatively clean. And so our second project focused the headwaters, and we worked with fish guts. So we wanted to know if fish from the headwaters had microplastics in their guts. And yes, they had. About 38% of all investigated individuals were positive for microplastics. And all these microplastics were fibers. The uptake was not species specific, and we didn't find any differences between feeding gills. On the figure below, you see the comparison between the benthic gill and the watercolor gill. It's very exactly the same. But NP abundance was correlated with Trichoptera abundance in the guts. Well, this means that fish which consumed Trichoptera had a lot of microplastics in the guts. And most probably this is related to the protective cases of the Trichoptera, which include microplastics in the construction of their protective cases. So if fish consume these invertebrates, they consume the microplastics with them. So where does this microplastic come from in the headwaters? Well, most probably they come from washing machines. A lot of houses which are constructed aside this the small rivers and streams, and they discharge their sewage directly into the stream. And with this discharge, a lot of microfibers reach the stream and they are discharged. So this is a study of the civil engineering department, which shows the quantity of fibers released in relation to the washings. And the tissue which released most fibers was polyacrylic tissue. So, and the last study which I'd like to present you is the occurrence of microplastics in liver and muscles of a detrivorous fish of our watershed. This is a very abundant species. And this study is currently underway. It's by Daniela da Cunha da Silva, which is my doctorate student. And she asked if microplastics pass the intestine-blood barrier, because we know that a lot of fish have microplastics in the intestine. What are the microplastic size distributions? And what are the concentrations in both tissues? And are they correlated? So she has some preliminary data on liver and muscle, but she is investigating brain and kidney as well. So, as you might suppose already, or a lot of particle pass the intestine-blood barrier. 50% of the liver samples were positive and 72% of the muscle. The concentrations were higher in liver than in muscle. She found 150 particles per grams in the liver tissue of her fish species. Size distributions between liver and muscles were approximately the same. And below, you see some photos of the shapes she found in her tissues. Fibers were not very common. Probably due to their shape, they are not easily absorbed from the intestine. So these are my results. I thank you for your attention, and most probably further details will be coming. Thank you.
Thanks, Dr. Uwe, for your presentation. And I would like to invite everyone, all the speakers, to come back. It looks like our audience was a little bit shy, and we are a little bit behind in time. So just to wrap up our section, I would like to thank you all for your participation. I think it was a very interesting topic for us for discussion. I personally didn't know much about microplastics, and a few things came up to my mind, so it won't be specific questions. So I would just open, it's more like a comment, and I would like you to make a comment on it if you wish. So one of the things I was thinking, listening to all your presentations, was that it looks like microplastics are everywhere, right? They are in the water, they are in the fresh water, they are in the marine environment, they are in the sediment. So is there the storm water? Is there any way of creating barriers to avoid the propagation of microplastics? Because once they are in the environment, they go to the organisms, and the organisms accumulate them. And then we have all this propagation of the effect. I was also thinking about the watershed effect, right? Because I don't know, one of the presentations I think mentioned that the headwaters would be in a little bit better condition than the downstream, which makes sense if you think that like a catchment accumulates all the water and drags down to the coastal area. So is there any physical barriers for it, or even any chemical barriers that we could think about? Or maybe we don't know that yet. I don't know, anybody? Please go ahead.
Well, I guess the solution of this problem is not technical, it's just behavioral. We have to diminish the input.
Yeah.
If microplastics and nanoplastics are already in the system, it's extremely difficult to remove them.
Right, and for a long time, right? Since they don't disappear.
Yeah.
And what about the potential of bioindicators? Because, for example, I saw like macroinvertebrates, and I am aware about some macroinvertebrates that can be used to indicate like water quality and categories of quality of water quality. Are we close to have like a biomarker for, or a bioindicator for microplastics, or perhaps it's too early?
I can sort of say something there. So I think the work that was done by PhD student Bing showed definitely here our little shrimp could be a bioindicator of recent microplastic occurrence in waterways. But potentially, like looking at the other work, you know, trichopterans, maybe it just might be they selectively take up certain things, like the shrimp like blue plastics. So potentially animals can selectively take up certain things. That's the only thing with them.
Yes, I think that's a very good way ahead. Try to find bioindicators in all communities, plankton and benton and fish. This is a way, but we have a black box. That's the effect of the microplastics on the living of all the fauna and human. This is a black box for us. And I think that we need methodologies to measure and to detect, including in our body. And there are many discussions to measure in the blood, in the components of the body. So we have to go fast, trying to find.
Christina, we think that fish are a model for other vertebrates, including humans. And if you follow that line of thinking, there's not much argument to say that if we find microplastics in fish tissues, fish muscle, liver, whatever, well, most probably you'll find them in humans. And well, the first publications are already appearing that show microplastic presence in lung, blood, well, probably everywhere. Right. And I don't know if you are aware of a Swedish publication and from 2016, which followed nanoplastics in the food web, and they analyzed fish, and they showed that fish which had microplastics in their brain showed differences in behavior in relation to the control group. And if you follow that part of thinking, you may, well, think of humans having microplastics in their head and changing behavior.
That sounds like horrible, horrible news.
Oh, well, you'll get used to it, I guess.
Oh my goodness.
I'll have a comment on that. I think that's one of the key things. A lot of research into actual impacts that microplastics are having, it's lab-based, and it's using very, very, very high concentrations to get an effect. We don't really know what levels in the environment actually cause impacts, and whether they are causing an impact.
Yes.
And that's a key area we need more information on.
Okay, and to finish it off, one last one I was thinking, if we can't avoid them, can we manage? For example, I think two of the presentations today spoke about the whole of water treatment plants and then constructed wetlands. And I was thinking about the natural wetlands as well. So what is the potential, for example, for us, okay, drinking water, right? So let's think about the drinking water. What's the potential? Once the water is treated in a water treatment plant, or if we have series of wetlands along the way to the water treatment plant, could we reduce the amount of microplastics in the drinking water at least? Could we reduce it like significantly? Or maybe not. I don't know. My impression was that water treatment plants were more efficient than the constructed wetlands. But I don't remember if anybody mentioned about some natural wetlands. Do you have data for that already? Or maybe not.
I have done some work, and I feel that wetland is not that effective. It's depend on if it is a new one, constructed wetland, maybe they can work, maybe after, again, it's type of microplastics. Overall, they are not that effective.
Right. So I open to you guys. Sorry, go
I was
ahead.
going to say, I think the key is just reduce plastic use.
Yes, yeah.
Right?
Yeah.
That's
Yes.
the biggest
Clear in
message.
front of us. If we have like a take-home message today.
Yes, and special for children, because we receive children with plastic. Everything with plastic. Oh, it's terrible.
So true. So would you like to invite you guys to give like your final say for tonight, and then I will wrap up the section. So please.
I think for me, happy to collaborate. If there is anything come across, please let us know. Related to treatment aspect, I think we have Jackie and myself, we are happy. I think that
Yes,
would be okay as
definitely.
well. Yes. To see how we can work together.
Awesome, awesome. Please go ahead, Jackie, if you want to give your final
Yeah,
words.
as Biplop said, if anyone's interested, we would be happy to look into collaborations. I have the work also with passive samplers, looking at pesticides, if anyone's interested in that. And use less plastics. That's probably the key message from today.
Christina.
Yes, I think that we have some connections regarding indicators of microplastics. I think that's a good way to begin. Perhaps a collaboration in several communities, natural communities, that can or could reveal in a better way the presence of microplastic. Will be a nice work together.
Yeah.
We?
Well, I found it very interesting that even in different continents, we think the same things and working on the same problems, sometimes in a very different way with different methodologies. But the scientific logic seems to me the same. And while I was very pleased to see the presentations of my Australian colleagues about the same subjects we were working here. And yes, it would be very interesting to think about a cooperation, especially with your area, Jackie, because I see we have very much in common with our research interests, and perhaps we're able to sort it out something.
Yeah,
That's awesome. That's awesome, guys. I'm glad to hear that, because this is like the main aim of these workshops, right? Is to foster collaboration between our groups, thinking that we have some global problems now, and the sooner we start acting, the better. And if we can collaborate, we definitely should start doing it, and fast. As much as possible. So I would also like to say to the audience to please don't be shy, contact our speakers, because there might be a PhD opportunity for you, there might be a chance to collaborate to some project or even volunteer, I don't know, but there is opportunities. It's just a matter of knocking the doors. And here we have this opportunity today. I would like to thank all the speakers for coming here and for spending this time with us and our audience. And thank you, everyone. It was great to have you here. And I'll see you in the next workshop.
Thank you.
Yeah.
Bye-bye.
Thank you.
Thank you.
Thank you.
Okay. Bye.
Bye.